Hey guys, in our last video for chapter 16, we'll talk about some of the finer points of replication. So in the previous video, we talked about the mechanics and the enzymes that are involved with replication and the situations where this occurs, only in preparation for cell division. So we also can learn a little bit about how we go about ensuring that the DNA is a perfect or as perfect as possible copy, how we can fix it if something goes wrong, and then we'll get to that end replication problem that we've been foreshadowing. Okay, so let's start with proofreading and repair. So DNA is kind of a beautiful thing. We saw the way that it could use its parent strands as a template for new daughter strands. Okay, but sometimes polymerases make mistakes. Okay, these are what we would normally call mutations. Okay, and sometimes mutations are beneficial, sometimes they're neutral, and sometimes they're deleterious, so they can have really bad effects. Okay, so we do want to have a little bit of flexibility. It's actually evolutionarily in our best interest that it messes up once in a while, okay? But for the most part, we really need it to do its job just spot on, okay? So to help with that, DNA polymerases can proofread. They actually can sort of proofread their own work as they go. So in terms of being a polymerase, they only synthesize in the 5' to 3' direction. So this means that they have 5' to 3' polymerase activity. They can only add to the 3' end. So we spent a lot of the last video talking about that point, okay? But if they make a mistake, let's say that they pair a cytosine with an adenine, they can backspace. Okay, so this is really only relevant to freshly added nucleotides. It can't just go back and proofread everything it's ever done. This is kind of like if you make a typo, you catch it, you backspace, and you fix it right there. It's not proofreading the whole document. Okay, so we call this, because removing a nucleotide, you can think about that happening, exonuclease, exo taking it out, okay, exonuclease. That occurs backwards. That's a backspace, and that's why we talk about 3' to 5'. So in terms of adding nucleotides, always add to the 3' end. Okay, so unless we discover some kind of new species or alien type of thing from another planet that does a different thing, we're always going to polymerize by adding to the 3' end. Okay, we're always going to do that. This exonuclease activity is just a backspace, like, whoops, messed up, back up, fix it, and keep going in the 5' to 3' direction. So that's one way we can ensure fidelity of the DNA sequence. We can also have some mechanisms that can fix things that happen either during repair that don't get caught, or that can happen afterwards. So mismatch repair is a type of DNA repair that does what you think it would do. It repairs mismatches that either happen because polymerase missed it, or because of some chemical reactions that can happen in the cell, sometimes just naturally, okay? So that can happen. Nucleotide excision repair is another type of repair that can replace other types of damaged DNA, okay? So this is actually, even though it'll seem a little bit detailed, we're talking about two different types of repair beyond DNA polymerases proofreading. There are actually several types of repair that repair different types of damage. There's some overlap between the two. It's an incredibly complicated thing, but this is just to give us kind of an overview and example. So let's start with mismatch repair. So mismatch repair is responsible for fixing simple mistakes that DNA polymerase 3 may make during replication, or again, as I mentioned, sometimes nucleotides can convert to other types of nucleotides just because of chemical reactions that happen in the cell. Some of it happens, like you may think, if you're in contact with a certain compound or it's environmental, but some of it's just naturally the types of chemistry that happen inside a cell for it to be alive. Okay, that just happens sometimes. Either way, we end up with a mismatch. We have an A paired with a C, or something's wrong here, and that will actually create a sort of distortion in the DNA strands. Okay, so those hydrogen bonds are placed the way they are for a reason, giving us an even width of that helix. Okay, and this isn't actually wound like a helix, but you can go back to some of those pictures. DNA is a helix that we've had in previous slide sets, and note that it's an even width. Okay, having something where you're not going to hydrogen bond together distorts that width. Okay, so we have enzymes that can roll along and actually find those distortions, those mismatches. Okay, so here are mismatch repair. We've got two main guys shown here, but sometimes there are a couple other things happening in the background. Mut S is proteins going to scan. I think of like S for scan. Okay, it's going to scan along, kind of like a mall cop looking for trouble. Okay, scan along and find this distortion, this mismatch. Okay, when it does, it calls in its friend Mut L, which you can think of as like for loop. Mut L will help make this loop. Okay, and Mut L in combination with some other components that can show up can actually cut the phosphodiester bond of this backbone. So we just cut it. And then the DNA is resected, so it's actually like exonucleases or, you know, something that will remove the DNA will come in and remove it, you know, prior to this mismatch. Okay, after that, the DNA is repaired. Okay, so we can have DNA polymerase come in and fix that and ligate it back together. Okay, so our DNA polymerase fill this in. Okay, and this isn't labeled, so I'm going to just say that it goes to the right, and then we seal up any nicks with ligase. Okay, so this is one thing that can help us repair problems in our DNA. Nucleotide excision repair repairs other types of damage. Okay, and the really common one is damage that is induced by UV radiation or being in the sun too much. Okay, so something that can happen when you have too much UV exposure, okay, either from the sun or whatever else you're doing that might involve UV, what can happen is if two thymine nucleotides are next to each other, so you know the sequence would be like whatever, whatever, T, T, whatever, blah, blah, blah. If they are next to each other in the sequence, they can actually form covalent bonds together. Okay, this is the nitrogenous base, not the backbone. The backbone is supposed to be sealed together. These are like gluing the two nucleotides together where they shouldn't be. Okay, and again, that can create a distortion, and this can result in problems downstream because this polymerase, if it tries to replicate this using this as a template, it doesn't know. Like, it's like, what is this? Okay, do I put two nucleotides here? Do I only put one? Do I put three? Like, what is going on? Okay, so we got to fix this if we don't want to propagate mutations downstream. So no, none of the nucleotides have changed, but because of this distortion, we're going to have problems if we leave it here. Okay, so we can have nuclease come in, kind of nick on either side of this. DNA polymerase fills it in like last time. Anytime you're creating new DNA, it's a DNA polymerase. Okay, so we're just going to adjust ourselves to that, and ligase can seal it together. Okay, so this is a popular one to talk about because this is one of the types of damage that UV light can induce. Okay, it can also cause breaks in the DNA, just straight up breaks, and a lot of other things downstream from that. But when you're in the sun too much, this UV induced damage, this actually can result in such extensive damage that your repair mechanisms can't even handle it. That's when your cells undergo apoptosis, they die, and you can peel off your sunburned skin in one large glorious layer. Okay, so that's the result of so much damage that those cells couldn't repair it, and they took one for the team and they went ahead and did apoptosis. Okay, so thymine dimers are just one of the types of damage that our nucleotide excision repair can come back in and fix. Okay, the people who don't have functional or good nucleotide excision repair often can end up with a lot of instances, of skin cancer just from being in normal sun exposure. Okay, so it's very important that we have this. All this stuff's really going on in the background and we had no idea. Like we just thought we were living our lives. No, your cells are constantly protecting you from natural things that may happen and dumb decisions. So thank your cells because that's what's happening. Okay, so we've got two types of repair that we'll talk about here. Again, there are many types, but these are good examples just to kind of give us an idea. Okay, if a mistake slips through, maybe it'll be beneficial, it may be neutral, or it just, may be deleterious and that's not great. Okay, so the last thing that we'll talk about is the end replication problem for linear chromosomes. So I've been hinting at this since the first video. Okay, so this is our linear chromosome, and this problem is going to happen solely because of the directionality of DNA, that leading lagging strand thing. Okay, so again, we're up here in the fork. Just ignore all this for now. Let's calm down and look what's up here. Okay, so our leading strand, here we go. We can just copy all the way to the end. Okay, so you can see here we've just copied all the way to the end. That's what's happened. The lagging strand, however, was dependent on putting those primers and then synthesizing in the right direction, the right hand direction. Okay, so let's look in this box. That's what's going on here. Here's this primer, Okazaki fragment 1. We put down our primer right at the end, Okazaki fragment 2. All right, we know that we have to remove those primers. DNA polymerase 1 does that. This one here gets repaired, sealed together. But what do we do now? Anybody got any answers for this problem? Okay, if you do, then you should probably patent that because there's not a good answer. Okay, we remove that primer. We cannot add on to the 5' end, so we can't just fill it in. There's nothing to template onto out here. Can't just float a primer out here and go. Well, shoot. Okay, we've been able to finish this guy, but not this one. Over time, that will result in shortening the chromosome on one end every time the cell replicates. So every time it replicates its DNA and divides, there it goes. Okay, so this is a problem because we have a little bit of a buffer on the ends of these chromosomes that I'll talk about on the next slide. But you can't just eat into your chromosomes forever. At some point, you'll cause damage that's going to affect the cell badly. Okay, you may either eat into more important information, you may cause other types of damage. Okay, it's going to be bad if we continue to shorten. Okay, but inevitably, this happens every time the cells divide, which depending on the cell type can be quite a lot. Okay, so your skin cells constantly dividing, your epithelial cells of various places constantly dividing, like sloughing off. Okay, so every time it's getting shorter. All right, so there's something to think about while you're trying to sleep at night. Okay, but what can we do? Let's first talk about the ends of these chromosomes. Okay, so they're not just ends. Okay, they have very specific structure. Okay, so this is kind of like leading up to the end, and this is the very end, and you can see it's got an overhang, just like kind of pictured. All right, this is going to be a larger overhang. The ends of the telomeres, or the chromosomes, are called telomeres, okay, because they're at the end. The telomeres are formed from short repetitive sequences, okay, so this one's like 6 nucleotides repeated, TTA, GGG, TTA, GGG. Okay, you don't have to memorize that. It's different a little bit for every species, but about 6 to 8 nucleotides is normal. And in our cells, and like most eukaryotic cells, maybe there are some exceptions, but in general, if you're going to think about humans, they're at least several 5 to 10,000 nucleotides long, okay, which is going to be good because it's going to spare us from some damage for at least a little while, okay. So this is the straight end of our chromosome. It's got the same thing going on at the other end. There's an overhang there too, okay. So these guys, these telomeres, this is straightened out, they actually loop back, okay? And all these blobs, these are protective proteins. You don't need to know. I've blocked out the names of them so you don't even have to worry about it. So just kind of focus on the structure, okay? So notice this red part here. This is that overhang, okay? This part here, this is right here we're looking, okay? This actually forms a loop that tucks, it actually tucks right back in itself, kind of forming, if you think about like the hem on your clothing, like on your shirt, kind of looped back on itself, and this is called a T-loop, okay? And the purpose of this is to kind of protect the ends of the DNA, okay? But no matter how looped back this is still gonna shorten every time, okay? Every time we replicate, it's going to shorten, okay? So we got to think about what that means, okay? So for most cells in your body, there's nothing that we can do about it, okay? Telomere shortening is actually thought to be quite strongly related to the aging process. So that's just how it is. Telomeres are going to shorten. There's no way behind it. But how do we have offspring for multiple generations, generation after generation, if the telomeres get shorter as you go? Okay, so we do have at least one solution for that, okay? And that is an enzyme called telomerase, okay? I know we've kind of become accustomed to the ACE, meaning it's gonna cut something. Not here though, okay? Telomerase will lengthen telomeres, all right? So telomerase is technically what we call a ribonucleoprotein complex, which means that it is a protein complex, lots of proteins together, protein complex, that's got some RNA with it. Okay, it's a ribonucleoprotein complex. Okay, we've seen an example of that before, the ribosome. Okay, so it's a ribonucleoprotein complex, which means it's got proteins, such as this purple part, and RNA, which is going to be this pink part. Okay, so this telomerase RNA, in this case, it's about 450 nucleotides long, okay? And it does not code for anything. It is not going to be a messenger RNA. It is a telomerase RNA. And what it does is help make a template where we otherwise couldn't put one. So when we couldn't really put another primer out here to help keep replicating, we can take this protein and it's got a piece of RNA and we're like, oh man, I could use that as a template and continue the leading strand direction, okay? We can't add on to the 5' end of this bottom strand. It can't happen, okay? But we can lengthen this part of it so that when we go to put another primer replication next time, we can put it further out here, okay? So we've actually indirectly lengthened that, but we can't just start adding on to it. That's the problem, okay? So telomerase does not stop telomeres from shortening. Telomeres is gonna shorten, okay? That is just the end result of having sticking linear chromosomes. It's gonna happen, okay? Telomerase can lengthen them so that we have, think about it kind of like an hourglass as these shorten replication after replication. If we add on to it, we've got a larger buffer so that we can go ahead and shorten them for longer, okay? So in our cells, in human cells, in most of the cells in your body, telomerase is not actually functioning in your adult self, okay? It is not on in your skin cells to speak of. So those telomeres are just shortening, man. That's just part of life, okay? But in your germline cells, the ones that will produce sperm or egg, telomerase is active in those because it lengthens the telomeres so that offspring born from those gametes will have the same length telomeres that you started your life out as. Once they're conceived and born, every cell division, including the ones that happened, the very first zygote, all of that, their telomeres are shortening. Okay, but they just give them a fighting chance by elongating them to begin with. Okay, and this is something people really talked about telomerase for a long time as kind of a potential fountain of youth because it is so heavily connected to aging, the aging process. However, it's not really feasible to just turn on telomerase in your cells. Okay, one problem with this is that it makes your cells kind of immortal, which sounds neat on paper, but there's one type of immortal cell that you don't want, and those are cancer cells. Okay, so cancer cells are immortal, and the reason they are is because they don't die when they're supposed to. Okay, they don't die when they accumulate huge amounts of mutations. They don't die when other cells are telling them to stop growing. Okay, their telomeres don't shorten at the rate that other cells would, and if they do, they just probably fix it in like a back alley way. Okay, so telomerase can also contribute to cancer, so you have to balance out what's happening. And I think for now, what we should probably all do is just be grateful for the telomeres we have, okay, and that we can pass on elongated telomeres to our offspring. So this is the end of all of our replication videos. So in all of them, we've covered the history of DNA as a molecule and how it was determined to be the hereditary information, what its structure is. Talked about semi-conservative replication and the mechanics of that. And then we've talked about some of the finer points, proofreading, repair, and the end replication problem in this last video.